Hello, welcome everyone. We're going to get started in just a few minutes. We're going to give you some time for everyone to join the webinar. I can see people are starting to come in. Welcome, you're all very welcome to this BioDT webinar. Today we're talking about digital twinning for harnessing genetic wealth in crop wild relatives. You're all very, very welcome. My name is Joanne Ahern. I'm part of the communication dissemination work package, and in the background I also have my colleague Rita Jufrida. You'll see her being very active in the comments. The chat and both the chat and the question and answer chat are both open. If you have any questions during the webinar, we'll be collecting them and answering them at the end when we have a discussion. And today with me I have Desalegn, Chala, and Zakaria, Kehel, and I'm just going to share a brief presentation from my side first, and you are all very, very welcome. And I'm going to go share my screen, and you should all be able to see this now. You're very, very welcome. As I said, we're here for the digital twin for harnessing genetic wealth in crop wild relatives. And I have here with me, I'm just going to speak very, very briefly about the BioDT project and give you some of the statistics for who has registered for the webinar today. And then I'm going to ask Desalegn Chala from the University of Oslo to begin by telling us about the modeling the distribution of germplasms of interest tool. He's going to give a brief demonstration of this, and then Zakaria is going to give an introduction to the focused identification of germplasm strategy tool. So it's going to be a very, very interesting hour. And at the end, as I said, we'll be having a question and answer session with our two speakers. You can add all of your questions in the Q&A box, and please, I hope you do. Please ask questions. This is what we're here to do. So just to give a bit of insight into who has signed up today. Today we're welcoming to the webinar experts from several different institutions, but primarily there are researchers who have signed up, individuals from biodiversity research infrastructures. As many of you know, we have 4 research infrastructures involved in the project. There are some people from industry and a few policymakers, and you're all very, very welcome. I hope you enjoy today. Then in terms of the distribution, we're from 17 different countries, the majority of which are in Europe, which we found very interesting again. And I just wanted to let you know that there is a hackathon happening soon in Oslo. And while it's a little bit late to be registering, we will have more information on the outcomes and the outputs of the hackathon, which is a bring your own data end user workshop. So it is a very, very exciting hackathon, and we're really looking forward to it. And if you want to be kept informed of these kind of events and these kind of things that are going on constantly in the project, I suggest that you sign up for the BioDT newsletter, as we generally keep that. We send that out every quarter, and we let you know well in advance. So please do that. And also, if you want to learn a little bit more about BioDT, we have a great deal of information on our website, but we also have all of the past webinar recordings are on our YouTube channel, or else you can interact with us both on LinkedIn or Twitter or X, whichever way you prefer to refer to it. And that's my little bit said. And with no further ado, I'm going to hand over to Desalegn. And thank you very much.
Yeah, thank you, Joanna, for your introduction. My name is Desalegn Chala, and I'm a researcher in Oslo University. My background is ecology, and I am working in one of the use cases in the BioDT project. BioDT project is a Horizon Europe project, and it has 4 use case groups and 7 use cases. And out of these, the crop wild relative is one of them. So I am the leader of this use case. Today, I will try to give you a brief introduction what a digital twin is based on my understanding. That means the definition of digital twin by drawing examples from different fields, including life science.
Perfect. Very good.
Okay. Sorry for the technical problem. So
No problem.
yeah, I was here, I
There
think.
we
So I
go.
will try to introduce about the crop-wide relatives, what they are, and then what crop relatives are and why crop-wide relatives, and then the MoDGP tool and what does the MoDGP tool perform. Normally, a digital twin is a digital counterpart of a physical object or a physical system in real time or in near real time with certain frequency and fidelity. That means we have the real space and we have a virtual space. And this real space and the virtual space are connected to one another via data stream. For this, a good example can be weather prediction. Meteorologists are simulating the Earth's atmosphere and predict and provide weather information in real time. The weather information for any location on the Earth is available through web services and smartphones. So that means the weather is a physical system, and what we access through the web service is what you normally call the digital version of it or the digital twin of it. I was just checking the weather here in Oslo. As you can see that it is negative temperature. Snow is falling, and at the same time, you can see the weather in Addis. The sun is shining, and the temperature is at a room temperature. So, I mean, like, to know about this information, like, one shouldn't have to be in Africa or one shouldn't be in Oslo. Like, you can just digitally access the weather information, and that means you are getting the weather information in a real time from a web service or from your smartphone. And that's a perfect example of digital twin for me. Again, we can also think of the smart home devices, such as the fire alarm, which is continuously detecting the concentration of smoke in our home and informing the fire department when the smoke is above a certain threshold. That means the fire department is getting information in real time. So that can also be a good example. In addition to that, we have satellite images which are orbiting the Earth with different temporal resolutions and having different spectral resolutions. Now it's possible to access these satellite data to analyze them and, for example, monitor forest disturbance in near real time. You can monitor forest disturbance due to illegal logging. It can be due to forest fire and so on. So in such a way, you can get information in near real time, in near real time. For this, for example, you can check the Luca app, which is, I think, updating the forest disturbance in 2 weeks' time, if I'm not mistaken. That means, just like you can digitally monitor what's happening to the forest ecosystem throughout the globe, be it in tropical forests, be it in Amazonia, be it at the backyard of your home. You can get information from the Luca app, which is updating in about 2 weeks, I think. The same principle can also be used, for example, to monitor farmlands from the time of sowing all the way to the time of harvesting. We can automatically access this satellite data, generate different kind of indices, which are indicators of abiotic and abiotic stresses. As soon as they are appearing, we can also analyze and get information on crop maturity and the readiness to be harvested. Not only getting this information, we can also create a kind of alert so that we will be informed about the status of the crop. So that's a kind of digital twin which is informing us in a near real time. That means it's the program which is doing this. So the program will be automated in such a way so that we can get information and we can get an alert. So we can have a digital version of that thing. Of course, what we are getting from satellite images is spectral information. Diverse spectral information from certain farmland means variety of plants in that farmland, probably indicating a weed, so that we can get an information and we can make an intervention. So we can digitally follow what's happening to our farmlands, and we can automate in such a way that we can get an information in a near real time. So in general, like, we are producing a lot of data. We have a lot of satellite images, and we are producing a lot of hyperspectral data, which can enable us identify plants at species level. We are producing a lot of sound or acoustic data, which can enable identify animals at species level. And we have a lot of molecular data out there. And so that means one can develop a program that can access and analyze this data to inform the state of biodiversity or nature in real time. And because of that, I personally think the time for transition to biodiversity digital twin is ripe. So coming to the DT, digital twin for me is all about automating data flow, fusing data from distributed data sources, automating regular model runs by feeding in updated data, followed by new model iteration. In such a way, static models such as species distribution model can be dynamic. And this also will enhance the number of model targets and the robustness of the model. That means with improved data, we can learn model for more species. And with improved data, the robustness of our model will also improve over time. And it also creates an opportunity to provide automated alerts for new data with predicted traits. For example, desired alerts in case of crop-wide relatives. Coming to the crop-wide relatives are wild plant species which are closely related to the cultivated crops. By the broad definition, it refers to all taxa within the same genus as a crop. And it also includes wild population of the same species as the cultivated crop. And for this, a good example is the coffee Arabica. And the coffee Arabica is native to East Africa, but it's introduced to different parts of the world. In its native range, we still have coffee in the forest, which is growing in the wild. So the coffee which has never been cultivated, which is naturally growing in the forest, so it's belonging to the same species as the cultivated ones, still it makes a crop-wide relative. The crop-wide relatives are highly diverse, and they are making about 21% of the world flora. That means if you come across, like randomly come across 10 wild plant species, the chance for all of them, 4 of them to be a crop-wide relative is there. I mean, 2 of them will be there. Or if you come across 20 wild plants, the chance for 4 to 5 of them to be a wild relative is high likely. That means they are really highly diverse. So coming to the question of why crop-wide relatives, and if you see the cultivated crops, farmers are selecting the trait of their interest. And because of this selection, the genetic makeup is reducing over time. For example, if you see this one, this is the wild maize 4,000 years ago in Peru. But as the farmers are selected maize with bigger seeds, finally we are ended up in this kind of mazes throughout the world. That means through time there is genetic homogenization, and because of that there is loss of genetic diversity and some adaptive traits, which are highly important to cope with diseases, extreme conditions, and so on. And the second one is environmental threats are reducing the potential food production currently. Like each decade, the potential of food production is declining by 2% because of climate crisis, because of biotic and abiotic factors. And at the same time, human population is really increasing very drastically. So if you see the crop-wide relatives, they have been in nature since the time of their evolution, and they have been through several selection pressures against both biotic and abiotic stresses. Through selection, natural selection, they have developed adaptive traits to drought conditions, waterlogging. They are growing in waterlogging areas. Some of them are growing in very warm areas, and some of them are growing in very cold areas, and some of them are growing in very saline areas. And some are resistant to insect pests, and others are resistant to fungal pests, and as well as other diseases. That means they have untapped novel genetic resources. And the objective is all about bringing back these untapped genetic resources and the genetic diversity that we have lost from the cultivated crops. And that's one of the aim. And the second one is introducing novel genetic resources developed against the selection pressure of biotic and abiotic stresses. So these are the two main objectives. So for that, we are developing a new tool, which we called MoDGP, or modeling the distribution of germplasm of interest. And a MoDGP is normally dependent on species distribution modeling. That means it uses occurrence data and environmental data. And these occurrence and environmental data are combined through model algorithms, and finally to produce a suitable habitat. That means it captures both the actual and the potential distribution of the wild crop relatives. And MoDGP is set in such a way that it can automatically access and standardize data of crop wild relative from online sources. That means it can automatically access data. It can automatically run models and produce habitat suitability maps. In addition to that, it can evaluate tolerance for abiotic and abiotic stresses. It can evaluate the tolerance of different crop wild relatives to biotic and abiotic stresses. And for this, we are developing a graphical user interface. And the graphical user interface will allow you to select different crop wild relatives and to check the tolerance to different environmental factors. Actually, we are still developing and it's not yet finalized. For example, if you consider Latras, it is the genus which is containing the crop grass pea. Within the genus Latras, we have about 180 different species, which are wild relatives to grass pea. So literally, you have to able to compare what the habitat suitability of all these species looks like. Not only that, we are also thinking of including the abiotic stresses so that you can check which crop wild relatives are highly tolerant to these abiotic stresses. These abiotic stresses can be acidity, it can be salinity, it can be temperature stress, it can be temperature stress, and so on. That means by just sliding those bars, you can see the degree of tolerance. So it also ranks populations based on the range of tolerances. It can also access and rank accessions from databases. There are accessions of land races, millions of accessions of land races in different gene banks. It should also able to access this data and rank based on their tolerance so that breeders will know which one to try first. And it also generates geographic areas where those populations are growing so that plant breeders can go out and make collections. So it has a graphic user interface, so that end users can make different kind of decisions. At the same time, it is also set in such a way that it can be transferable across crops and the traits. That means it can work for different crops. It can also work for different traits. And it's also automated to regularly run with updated data and provide alerts on newly predicted traits of interest. Yeah, so that's generally what the MoDGP looks like. Finally, I would like to thank Doug Anderson, who is the co-leader of the use case, Thomas Rossi, who is helping with high performing computing, which is the Lumi. Thomas Martinovich, who is also working with the graphic user interface. Klaus Wieland, who is helping with making the data which we are using fair and making the output which we are producing fair. And above all, Eric Kusch, who is also helping with coding. With that, I would like to say thank you. And if you have questions, I will try to answer later on. Thank you.
Thank you so much, Desalegn. That was really, really interesting. Zakaria, are you ready? Do you want to share your slide? And
yes, I will.
Brilliant.
Yeah.
And just while you're getting ready to share, I'll just remind everyone that the question and answer box is open. So please put your questions there and we'll be asking our speakers at the end. Thank you so much.
Do you see my presentation in presentation mode?
Yes, I see it perfectly.
Is it moving?
Yes, it is. It's going between two slides. Yeah.
Okay. Thank you.
Thank you.
Thank you. So thank you for this invitation and thank you for the participant to be here. I want really to share with you some of the experience we have in really combining different data sources, including phenotypic environment and genomic to mine collections. Collections, I mean ex situ and in situ. And this is a really in the same context of the first presentation, what we want we want to go digital and look at how species are distributed, but beyond that, how to take advantage of what we have in different sources in terms of data to get the best. And here I will be looking at the traits again linked again to the previous presentation. So mainly we know that we have plant genetic resources collections. Again, we are talking about ex situ and in situ collections, and we have the elite material that we want to serve by looking at the distributions of species, to collect what we still collect or conserve, but also to use them and use this diversity in order to develop elite material. And of course, we know that we have the biggest diversity, looking at the size also for collections and occurrences, but we don't have a lot of data in terms of performance and in terms of characterization. There are a lot of papers, especially when it comes to traits. So we have some data in terms of distributions, especially linking it to what we just heard in the first presentation in terms of climate, soil, etc. But we don't have the performance data there, and since we need to mine them for further use, we are lacking this information, and we are trying to find methods to overcome the sources of information. So mainly this is how it looks a little bit. So we have different groups looking at the environment. We just saw the example. We have different groups looking at the evaluation. So different data scattered everywhere. What we don't have a system to look globally at this and take advantage of it. So the data is there. But we need, however, to turn this scattered data into useful information for us and for our users. So mainly what we have and what we know about the collections, we know that we have passport information. When we talk about passport information, or maybe the occurrences, we know that we have geographic coordinates, and those geographic coordinates, we can link them to kind of environmental characteristics or characterization in terms of soil and climate. Sometimes also in terms of socioeconomics and what's happening in that microenvironment. So we have some phenotypic information and evaluation. However, that's not for the full collection. We don't have that information because we have some users depending on project and the necessity of evaluating portion of the collection for specific needs. And we can actually take advantage of that information because we can't, and it does not make sense to go and evaluate all the collections to be able to target specific traits because it's just simply impossible resource-wise and in terms of what kind of traits we want to target because different groups and different users have different information. And of course, lately we have access of genomic information that we are hearing that more and more it's getting cheaper, even though it's not that cheaper for us working on plant genetic resources because of the size of the collection, even though the price by sample to sequence is getting lower and lower, but looking at the size of the collection, we cannot just go and genotype everything. So our ecosystem or data ecosystem to turn this into information for us and for users is really looking at the data availability and this data sitting somewhere, linking it together. So mainly here in this example, I'm looking at our documentation system, which is our database where we are holding information on passport, evaluation data, characterization in terms of descriptors, diseases, and other morphological descriptors. But also we can link it to all available or open data, open source. It's mainly open data, not open source, like environment and world claim and maybe the data that was shown in the previous presentation. We are linking it also to what we call breeding management system, where we have access to data that it's generated by users, sometimes breeders, that can help us mine those resources. And the genotyping component in case you have genotyping. And sometimes now we are looking at the nutritional value for plant genetic resources, and we can link that through API for people that are familiar with this or RAPI API to link all data. I mean here that one does not need to have one database to mine plant genetic resources, but rather link the available information out there or open information out there. So from this, looking at our pipeline and the ecosystem, I want just to mention this strategy that we have developed at ICARDA a couple of years ago, what we call Focus Identification of Germplasm Strategy Fix, which is really looking at principle of adaptation and evolvement or adaptation of plant genetic resources in their environment of origin, and we are trying to link the trait and environment associated with selection pressure with accessions. Those can be gene bank accessions ex situ, but it can be also an in situ accession still conserved in its origin. And for that, we are focusing on the accession that most likely have developed adaptation mechanism and have that specific trait variation. And we have here, of course, two methodologies that we are using. One of them, when we don't have access to any information on that trait, that means somebody is asking for resistant to X disease and we never evaluated for that disease. So we use what we call philosophically filtering methods, and I will show an example in the demo where we look at really what are the ecology of that trait to have adaptation. And once we have some evaluation data for portion of the collection, we use what we call the predictive modeling methodology, where we use machine learning algorithms to link and find the function between the trait or adaptive trait of interest and any other characteristic of site of collection, but most precisely talking about the environmental characteristics in terms of soil and climate. So those are really very clear pathway, but they are connected together because once we evaluate some portions based on the filtering method, we can go and apply machine learning methodologies. So this is how it looks, of course, when we find a good accuracy of a machine learning algorithm. That means there is a tight relationship between environment and the trait of interest. Once we are happy of the performance of the machine learning, after different or several tuning, the best model is used then to do what we call predictive characterization and assign a probability of success of a given accession to a trait or adaptive trait of interest. So we have developed an R package, for example, where one can get access to collections and passport information. We can get access to different infrastructure in terms of climate and environment, but then run all the machine learning algorithms that will help us assess this tighter relationship or not between the trait and climate. So this R package is available, of course, in the CRAN, easily and freely accessible, that we can test, and we will be happy if you test it and get feedback about it. However, since we know that a lot of users, including plant genetic resource specialists, etc., they don't have, it's very difficult to use those software like R, which is coded software, and if you don't use it every day, it's kind of difficult to go back after a couple of weeks and use the same thing. So we are keeping this one and we are improving it for specialists. However, we are not forgetting about our users, that they are not really familiar with statistical software and coding software, and of course we are developing a graphic user interface that I will show in a minute. So of course, this is just to give you an example of pipeline for machine learning algorithms where we are testing several of them. And of course, without having a big knowledge of machine learning and good statistical knowledge, you can still run those methods, because we tried at our best to make it little user-friendly, and people don't need that deep knowledge of machine learning to be able to run them. So here I will show you a small demo, and hopefully if you cannot see the demo moving, please tell me so I can adjust. So this is our user interface that is really linked to that R package. For now, we have three models only, and people can get access by crop name, which is really access to our database at ICARDA, which is a really open sources and publicly available. You can upload your external data, which is a CSV file with the coordinates, because we need the coordinate to get access to climate information. So this is how it looks like. You can, of course, map your accessions to see the distributions geographically, but also do some thematic mapping, like population type, or to look, if you want really to look only on crop wild relatives, for example, then you can filter on that. Or maybe look at the taxon if you want to use or filter based on a specific taxon that you want to work with. Also, it can give you summaries about how your data and information is scattered in terms of country of origin, taxon, and species. So from there, whenever you have coordinates, you can extract whether freely available climatic information. Here we are using WorldClim, but we are targeting other climatic and also soil data, because that's very important, and a lot of people are asking for trait related to soil. Unfortunately, there are not a lot of resources in soil, and we will be using the FAO harmonized WorldClim harmonized soil database. So this is just a tool to look at the distribution of climate. And once you have the climate, you can do some filtering. We call it multivariate analysis, where one wants to look, let's say, accessions that may be for drought tolerant or heat tolerance, or accessions that will be performing better under certain conditions. So we have developed this module where the user can go and actually filter any kind of information we are extracting for the collection, and it will give you your subset that we will use, because we know that users, they don't want to deal with big numbers, and they want just small subset based on that filtering to maximize their chances to find the trait of interest they want. This is, for example, the subset we developed for temperature between the range of 4 and 16. You can do some clustering where a user wants to look at groups of germplasm based on temperature, rainfall, humidity, etc. And of course, you can do those statistical method, which is grouping or clustering based on the original data set or the collection, or you can use the filtered one that you filtered before based on your needs in terms of temperature. Another important one is really to be able to run principal component analysis. This is statistical method that will reduce the dimensionality. If you look at the climate, it's two huge data sets, and we want to see if there are patterns of summarizing the data set in the climate based on species. Are some species really, for example, evolving in specific environment or not? And from there, we can use what we call core collections, because they are still important for our users. Again, users don't want to deal with the big collections because they cannot evaluate them or characterize them. So you can use core collection that help you maximizing the diversity you have, and hence here we are looking at the climatic or environmental diversity in this example. So you can, if we have this collection of tens of thousands, you can reduce it to hundreds so the users can do it. This is an example whenever we have, we call it trait analysis, whenever we have access to evaluation data, where we can get access to it via API, linked to passport information. We can have, for example, in the case of ICARDA, the user can get access to the characterization and the evaluation information. You can summarize this data, summarizing it by having the mean, because sometimes we are evaluating in different environments or different locations. You have a couple of plots that help the users extract or select what they want, but also look at patterns in terms of spatial structure of the trait. Do we have a really kind of pattern that we can use later in terms of looking at the distribution of trait, etc. So this is where we are in the graphical user interface. We are still testing a model where, based on these traits, we can link in it to the climate that we extracted in the second module, and then run machine learning directly in graphic user interface, so the users that they are not really expert in modeling can use it and link it. And once the user finds a tight and strong relationship, then you can go and do what we call predictive characterization by assigning a probability of interest. So I want to add that we are improving this methodology of FIGS by adding a component of genomics, where we do what we commonly use as genomic selection or genomic prediction. Whenever we have access to genomic information, we link it to passport information, and we use both of them in a comprehensive method to increase the efficiency of predicting. So we saw that we can predict based on the climate, but we saw in one of the work that was published 2 years ago that if we add the component of genomic, our prediction is going higher. And then, of course, that showed a better predictive characterization for our collection. Quickly, I will just mention what we are doing as added value to plant genetic resources, including crop wild relatives, is that we can link the genomic in a different way to our passport information from the origin of plant genetic resources, so we can reveal regions that they are associated with the change on temperature, and hence we can look at those plant genetic resources and wild that they are maybe having some alleles associated with the change or increase of temperatures or a reduction of humidity or availability of water, and that's something that we are working at. However, it's still not at the level of production where we can put it in a graphic user interface, but the science part of it is very valid and very helpful in assigning or linking a trait of interest to to plant genetic resources. So yeah, what we want really to organize, curate, and connect different data sets to be able to do this kind of data mining. And that data mining, which is helping us from doing predictive characterization, since we cannot characterize everything, and increasing the predictability of by using the maximum of data we are having. So I want by this to thank you, and I want you also to share with you one innovation that we just developed based on FIG's methodology. It's you can have the QR code in the upper one, which is what we call substituting tool. You can have access to it to Genesis. Genesis is really a home for a lot of crop wild relatives and plant genetic resources, and you can test it. I'm also happy to receive your feedback, and I'm of course happy as well to answer any questions or comments from your side. Thank you very much.
Thank you so much, Zakaria. That was absolutely fantastic. All of the presentations and the recording will be available afterwards. So if you didn't manage to catch the QR code, you will be able to grab them afterwards. And with no further ado, thank you to both of our speakers today for your fascinating presentations. And we have some questions after coming in, if you're ready to take them. So I guess we might go in order of the way they came in. And I know, Desalegn, you've already started to answer some in the chat, but I'm going to ask it out loud again, just so that everyone can hear it. So this comes from Joanne. From which online sources does it allow to download and standardize the occurrence data from?
Yes, I was responding by the chat, and currently we are relying on GBIF, and we are also planning to use Genesis in the future. And at the same time, for the cultivated crops, we are also planning to rank the accessions. That means we need to access data from gene banks as well.
Okay, very good. And then to Zakaria from Magali. Apologies for the pronunciation of the name, Magali. Zakaria, could you please be precise about what EWAS is?
Yeah, sorry, I have put that we call it EWAS, but I think lately we have decided to call it environmental GWAS. I was typing the answer, calling it the environmental GWAS because to not mix it with the expression WAS, which is a really association studies like what we do in genome-wide association studies, where we link the variation in the environmental or origin of plant genetic resources to the variation or allelic variation in the genome. It's a really statistical methodology that we are using to form those associations that we think are linked to adaptation in terms of abiotic stresses, drought, heat, salinity, etc.
Okay, thank you. And then, okay, the next question is, could you provide examples of, if there are any, of course, of how digital twinning has been successfully applied in other agricultural projects? Projects, yes. And this is for both of you, so whoever wants to jump in.
Will you go, Zakaria?
I don't see the question.
Yeah, the question is like yes, they want to hear examples if digital twin has been applied in agriculture or not. I personally know that it has been applied in forestry, especially in monitoring forest disturbance. But for my knowledge, I know there are some experiments which is going on in Wageningen and so on. But on a bigger scale, I don't really have a good example. And at the same time, we have to mention that BioDT must be the pioneer, because the concept of digital twin was mainly there in industry. So it is quite a new field to biodiversity, and even it's quite a new field to life science. Because of that, I don't think that we can have a good example where it has been widely applied. So I will refrain. I mean, I don't really have a good example, but yeah.
But it's interesting also that there are no good examples that come to mind, but BioDT is leading the way in this. And actually, you know what, let's switch from the current to the future. One of the other questions is, what advancements or developments do you foresee in the field of digital twinning for crop wild relatives, or in agriculture as well? I'm going to expand this question. Like, when you're looking to the future from your research now to the future, what kind of future developments do you both see?
Yeah, again, it's about digital twin. So since I'm actively involving in that, especially I was talking about like satellite imagery, right? So we really have a lot of satellite images, and some of like the satellites have a revisiting time of 5 days. That means each 5 days they are taking images. And these satellite images have got a lot of data, and you can produce a lot of indices. And some of the indices are indications of like crop health. Some of the indices are indications of, water stress. Some of the indices are indications of maturity. Some of the indices are even indications of how much yield you can obtain. So especially by, creating like a program which can automatically download these satellite images, analyze it, and then generate, information so that you can digitally follow like what's going on. And there is a huge chance for that. And at the same time, like when it comes to crop wild relative, so breeders are not mainly using crop wild relatives in breeding. Zakaria may help me with that. They are mainly relying on like land races and their own breeding lines, but still there is a huge potential out there, especially what we are doing on a use case. I think if it is used by plant breeders, there's a huge opportunity out there. And sometimes I think I'm over optimist, but I personally think that it's something which can revolutionize knowledge production, and at the same time making a huge impact, especially in the agricultural section, if you can utilize it effectively.
Okay, makes sense.
Well, I think some of these technologies are already used in digital agriculture or precision agriculture, where you look in a real time on stress of the plants. So that have been used for years now, but also used in terms of needs of fields, in terms of fertilizers, etc. But also looking at prediction of yield before you go to the harvest stage. I'm talking about like just after emergence, looking at this real-time data that you can use in a context based on a history or experience from previous data, and you are taking real-time data to actually be able to predict your productivity months before you go to harvest, and that's in terms of monitoring, of course, the availability of commodities, but also in terms of stabilizing the price. What we are trying to do here is also used in biogeography and in animals, wild animals. That's something that it's used extensively. But for us here in crop wild relatives, again, for a long time, breeders, they were using land races and cultivated species, which in general have less issues with the availability of information compared to crop wild relatives. Crop wild relatives, there are several papers that one of the issue of using them, of course, there is the issue of using them because it's taken time, they're difficult to integrate into breeding program. But one of the issues is really the availability of information. So if we are going and using the modern tool of doing predictive characterization, that will have access to information about crop wild relative and also the opportunity of using them more and more.
Yeah, I think keeping going with this theme, like how do you see these tools also contributing to overall goals within Europe, but also worldwide, for sustainable agriculture? So you're talking about tools that you can look at the yield, you're talking about the tools that you can see the effect of fertilizer and crop wild relatives. When you're going beyond that and looking at other modeling, how can they contribute? And also, sorry, I'm also going to throw another question at you at the same time. which is, could you talk about some of the challenges that you've encountered or seen happen when implementing these tools in real world scenarios? Sorry, those are two very different questions, but
okay.
yeah.
Like, yeah, if I have to like build on what Zakaria was saying, like, I think he was mentioning about lack of information. So one of the main advantage of digital twin is its ability to access data from different sources, make them interoperable, fuse them, standardize them, and then finally run a model. At the same time, we are aiming to make it, like transferable across crops for any crop, be it maize, be it wheat, be it barley, whatever crop you mentioned. You just tell, like the tool which we are creating so that the tool can automatically generate, like depending on your interest, tuning into your interest. Like if you want to improve, let's say, drought resistance of barley, then it can tell you which crop wide relative is the best and where to find that population at the same time. So especially in making the information available, in making, like what population to try first, what population to try next. So that way it makes life very easy. Above all, especially the accessibility issue is really very important. So it is also user friendly at the same time, and that's the major contribution, I would say.
That makes sense. There's actually another question coming in from Joanna again. She's asking, how do you see these two tools contributing to the conservation of PG or?
I can go if you want.
Why,
you can
yeah,
make a
go
supplement
ahead.
on it.
Go ahead.
Normally, I think fix is mainly about like targeting the genetic resources, which are very useful, primarily very useful for breeding purpose. And MoDGP is more or less about that. There is a third tool which we haven't presented here, which is called Caffeitogen.
Okay.
And I think Caffeitogen is more about conservation. Sometimes like you may have a very ubiquitous plant, a very ubiquitous wild plant, and you may think that it may not be important to conserve it, but Caffeitogen is making an ecotype of it. That means it tells you how it is genetically diverse. And it tells you what it means to lose some population. Let's say if you lose a population from Norway, are you going to lose like the genetic diversity as well or not? So there is a tool which is mainly tuned toward this, but at the same time, like MoDGP also tells you like the distribution and in which protected areas these wild releases are occurring and whether they are protected or not, or indirectly you can get an information. But there is a tool which is specifically meant for this, which is called Caffeitogen, which we are using in the hackathon, but which we haven't presented it here.
And just on the hackathon, I know it's too late for some people to register now, but is there any way, will there be other ways in the future for them to share insights or register for other hackathons? Is there anything you want to say to that, Desalegn?
Actually, they have already heard from Zakaria and me. If there is an interest, I think there is a chance to make it at least online. So they may not physically attend it, but like as we can also see the response which we are going to get from the Oslo hackathon. If more people are interested, it's all about like all what we are doing is for the end user. So I think that's the biggest reward for us, and why not? Yeah.
Oh, brilliant. And Zakaria, do you have anything else you'd like to add?
Yeah, just for Joanna, I think as it was correctly mentioned that the fixed part is mainly for use. However, lately we were looking at conservation, and as you know, to use all those data modeling technologies in conservation, we do what we call gap analysis, which is really using this data to go and look for hotspots. Chala mentioned in his presentation of distribution models or distribution spots of species to go look for diversity that is not conserved in situ or ex situ. So what we are trying to do, we are trying to go beyond that step further and say, rather than looking at diversity at large, we look at diversity or variation of traits of interest. So we are using fixed methodology to do what we call trait gaps. And I mean, like, if we don't have access to diversity for salinity, for example. So we use fix and go and target collecting in the areas where we have more chance to find saline tolerance PGR, which has, you know, plant genetic resources. Yeah.
Thank you. At least I can definitely say I've learned a few things today. And
Nice.
we're definitely running towards the end of our time. So first of all, can I say thank you both so much? I know a lot of work went into both of those presentations, and I hope you enjoy the hackathon next week and that you have a great and productive few days. Apart for everyone else, these tools will be further explored both during the Oslo hackathon, but also afterwards. And maybe we might present them in another webinar in the second half of 2024, if I can convince these two speakers to come back. And thank you all so much. I hope you all have a great rest of your day. And remember, the recording will be available on the YouTube channel, and we'll get the slides up again as well after the webinar ends. And thank you both so much. Okay.
Thank you. Yeah,
thank you. Bye all.